Thank you very much for the invitation to be back in Singapore. It's a real pleasure to be here. I had an interesting week so far and looking forward to meeting particularly the young folk who are here. So, very different topic. I'm going to talk about DNA. I started out as an organic chemist actually, and one of the aspirations of chemists is to understand the relationship between molecular structure and properties and function. And we've just heard about that in the material world. I'm going to talk about biology. Now, the story I'm about to tell you is one of three aspects of DNA that my lab has been interested in. And this is an example of where, again, basic research has led to something that's had commercial impact and then societal impact. And this company, Solexa, which I co-founded with David Klenerman in 1998, this is where all the ideas and the proof of concept work we did in the university was reduced to practice into a commercial working system. And that system and the company, in fact, was acquired by an American company called Illumina. And so all the technologies that drive Illumina sequences came out of this. Okay, we're going to start with some basics. 71 years ago, we learned that two strands of DNA spend most of their time actually looking like this, a double helix. Now, DNA is a polymer. One strand of DNA is a linear polymer composed of 4 molecular building blocks that we abbreviate to the letters G, C, T, and A. And the big discovery 71 years ago in Cambridge with data from London was really about the informational aspect of DNA. DNA is an information molecule, and in fact it has bigger dimensionality in terms of information than what I'm going to tell you about today. But one aspect of the information comes from the way these letters, now I've drawn out the structures of, we call these bases. The C base communicates with the G base through very specific geometrically directed hydrogen bonds shown here. And similarly, T connects with A in this sense. So if there is a T in one strand of the double helix, there's an A in the opposite strand, and similarly for G and C. Now, this constitutes a mechanism for transferring information from one strand to the other strand. And this is absolutely fundamental to biology. It is a reason why DNA can be copied and replicated when cells divide. It's also the molecular basis for what we call the genetic code. Now, so you can think of DNA as a linear sequence of 4 bases. So one of your cells has 2 copies of what we call a genome, and one genome is about 3 billion letters arranged in 23 parcels called chromosomes. Now, collections of letters in regions form what we call our genes, and genes can carry the code for another polymer, and this is the polypeptide made up of amino acids that constitute proteins. So the protein code is defined by the DNA code. And proteins constitute the structures, the catalysts, and the molecular machines that drive life. In this sense, this primary code, the sequence of DNA, provides all the information for the machinery that constitutes cells and life. Now, about 2% of the genome does that, and for a long time the other 98% was a question mark, because it doesn't code for proteins. We now know that this non-coding part of the genome is actually important as well. It doesn't code for proteins, but it's important for regulating the genes that do encode for proteins. Okay, so I chose to work on DNA because there's a long and interesting history of DNA-related transformations in Cambridge. So it's a natural thing to think about. There are double helices all around the place, and actually it's sort of very much ingrained into the way a lot of people think. But more broadly speaking, determining the sequence of DNA is a sort of fundamental characterization of a living system. Now, for humans, this was first done by the International Human Genome Project, which was comprised of campuses called genome centers around the world. And the driving force for this project was really very fundamental. It was to define what a human genome looks like. It was just one genome which became a reference or a scaffold for other genetic research. Now, you need to know the genome sequence to understand really what it is that makes us who we are. You each have a genome that's unique. It's what makes you unique. So if we want to understand the basis for why we are all different, we need to understand the genome. And this leads naturally to understanding what genetic factors can either cause diseases or contribute towards a predisposition to diseases. So this is fundamental. So the International Human Genome Project provided the first blueprint for this. Now, what we were doing in the lab was nothing to do with that, actually. This slide is to introduce some key players in the project. So this is David Klenerman, who was actually in Singapore at GIS last year. Now, David is a physical chemist, and I met him in 1994, just after I joined the Department of Chemistry, because I was doing some experiments and submitted a paper, and I needed a laser to carry out certain experiments, and I didn't have one. So I went around the building, and one of my colleagues said, you should go and talk to this guy Klenerman. He used to be at BP, but he's just joined the faculty, and he has lots of lasers. So I went to talk to him, and we carried out the experiment. The paper got accepted, and then we kept talking. And David was looking into single molecule optical detection methods that were relatively new at that time. Now, I was interested in one of these molecular machines that's involved in replicating DNA, copying DNA. This is a class of enzyme called a DNA polymerase. It literally polymerizes DNA, guided by a template sequence. And we came up with an idea, and we wrote a grant, and it was funded. And the idea was actually very fundamental. It had nothing useful implied in it other than to learn about this process. And it got funded, and we took on a couple of postdocs. So Colin Barnes was an organic chemist in my lab, and Mark Osborne, also a physical chemist. So these guys were also key to the genesis of this project. Now, what we were doing is taking a piece of fused silica and coupling to it a piece of synthetic DNA. So the long strand shown here is serving as a template, and the short strand here is going to be extended by this molecular machine in this direction. And each step, these Watson-Crick base pairing interactions allow the template to tell the machine which of the 4 building blocks to put in each place here. And this mirrors what happens when a cell divides and the DNA replicates. Now, we built an instrument capable of visualizing single fluorophores in water at room temperature, and this was actually quite new at that time. And our reasoning for this project was new observation techniques drive inflections in science. If you can see things that you previously could not, there's a strong likelihood that you're going to discover new phenomena. And this reaction catalyzed by this machine, so fundamental to life, had to be important. So this was a driving force for this experiment. Now, one of the ways we did this involved taking these building blocks, G, C, T, and A, and attaching a fluorescent dye. And so you could literally watch the dye-labeled building block be incorporated by the catalyst as it extended this DNA. So we did this to really sort of probe the mechanics of this process with a new observation technique. Now, I'm here to promote science, not beer, but actually I joined the department in 1994, and the work that I've just been describing, we started in 1995. So it was literally one of the first things I did in my career as an independent scientist. And I should say, at that time, we were scrabbling around to try and find a big problem to work on. And one of the best pieces of advice is one of my senior colleagues and mentor gave me at the start of my career was, he said, you need to spend some time struggling to find a problem that's worth spending at least 10 years of your life on. At least 10 years of your life. And that was quite a daunting ask. And this is what we were doing. The significance of this place, now the chemistry department is behind this house here, so it's really proximal. And at the start of my career, and David started at the same time, we didn't have an office. Then after a while, I was given a very small office, probably the size of these 2 chairs put together, which I shared with another junior faculty member. So actually, if you wanted to go and chat, you had to go somewhere else. And we used to come here quite often to chat. I now have a great big office, and I don't seem to go there very often, actually. So there's some kind of inverse correlation between my office size and spending time here. But the significance of this is we were doing something, but we were actually still reflecting a lot on what do we want to do next. And it was during conversations here, and we bring our students here as well sometimes, that we were thinking about the Human Genome Project, because we could see a way to take what we were doing and adapt it in such a way that you can actually read the sequence of the DNA molecule that we'd pinned down on the surface. So we went and had a chat with the Human Genome Project people at a place called the Wellcome Trust Sanger Institute, which was nearby. And at that time, they were less than a third of the way through the project. And we just wanted to understand how things were going and to be educated a little bit about this project. But one of the questions we asked, and this project, ultimately it took about 10 years, thousands of people and machines. And I think the cost was about $1 billion, order of magnitude cost. It was admittedly the very first human genome, so the second one's a little easier once you have a reference. But it was expensive. And our question was, well, this is only one genome. We're all different. To really ask these probing questions, we need to be able to sequence on a population scale and start looking at the information from everyone. How are you going to do this? And there was no answer, actually. There was nothing on the horizon, and we were a little surprised that there weren't any major projects at that time to enhance sequencing. And so we shared with them some ideas and thoughts we had, and the message we got was this was a really interesting problem worth working on, and the world needed a better solution. Now, the adaptation of what I described is this. Here is just a schematic of a piece of sequence that now this is the template, and we want to read this template. We attach it covalently to fused silica, and there's a little bit of DNA here that we can call a primer. Now, the way these machines work is they catalyze a reaction with this oxygen here and incorporate a building block here. And the ingenious part of this is this T base tells the machinery to incorporate an A. And this A base will tell the machinery to incorporate a T. Now, this is very accurate. It has to be, otherwise our genomes would change every time a cell divides. Very rarely makes a mistake. Now, what we reasoned is if you could color code each of the 4 building blocks with orthogonal wavelength emitters, you could add all 4 and let the system decide which is the correct one to incorporate. Now, we had to somehow engineer a stop here to stop the second one from going in. If we then imaged this surface, we would see the color here and we would know which one has gone in. So we would read 1 base at this site, spatially on the surface. Now, if we could somehow then detach this dye and also detach the blocking group here that was preventing the next one from going in, we could go around the cycle a second time, and then we watch this T go in opposite this A, bringing in a different color, and go around a third time, and so forth. And at each step, you'd literally see a color telling you the identity of the base. So a series of color changes would give you the sequence. Now, this is very different to the existing method that Fred Sanger invented in the late 1970s. It was being used in the Human Genome Project. Now, this in itself, which is really sequencing DNA that's attached to a solid support, doesn't get you anywhere. But what it enables is a mechanism to parallelize the whole process massively. So this is really what our vision was. You could take DNA from cells, fragment them, and there are standard ways of doing this. You can use ultrasound, you can mechanically shear the DNA into smaller pieces. And we did some early experiments to show that you could monodisperse these pieces on a surface so that each fragment would sit in optically resolvable space on the surface. And if you then sequence these using the solid phase method I've described, each cycle at each site, you would see a color that would identify one base. And after n cycles, you'd get a read that was n bases long. Now imagine doing this on 1 million different fragments of DNA at the same time. 1 million features. You start to see possibilities to scale the sequencing approach. Now, this is literally a very simple order of magnitude type calculation that we did. We simply made some assumptions about the spatial distribution based on experiments. We knew about the kinetics of the enzyme that we would use. Now, the bottom line is, this told us that we could probably build a system that could sequence of the order of 1 billion bases of DNA in one experiment, on one system. Now, at that time, the automated machines that filled all the genome centers would typically sequence of the order of 100,000 to 1 million bases per experiment. This told us that we could probably go 1,000 times better. Now, importantly, because a human genome is about 3 billion bases per copy, we're in that sort of zone of being able to sequence a human genome-sized DNA sample. I'm going to come back to this figure a bit later, but it was just a guiding principle that encouraged us to keep going. Now, I'm not going to delve into all the technical details, but there was one deviation from our original plan, and that was instead of sequencing single molecules, we adapted a method that was created by a French molecular biologist called Pascal Meyer and his team in Geneva. It was essentially a method to take one fragment of DNA on the surface and amplify it so you have hundreds of copies of identical sequence, but they all occupy the same space as the original fragment. Now, what this gives you is it gives you a brighter signal and allows you to use optics that are a little bit simpler camera, a cheaper camera. It also potentially improves the accuracy, because if you have any stochastic error-generating modes on a single molecule, they get ruled out by all the other copies. Now, this is an image. This is real data, actually. It's my only data slide. And you're probably wondering what it is, because you see lots of little spots that are colored green or red or blue or yellow. Now, if you look at this expansion here, this is actually a CCD image. Now, charge-coupled detectors have been used for quite a while in astronomy. And they use a principle you heard in Martin's lecture, the photoelectric effect. And very good for scanning surfaces and getting images. They were also in the very first smartphone cameras used CCDs as well. So this is a CCD-generated image. And what you see here is each of these spots is a cluster of DNA molecules that was generated from one fragment at one stage during this sequencing process. And the color is telling us what the identity of the building block is that's just been incorporated. Now, you'll see that they're not regular. They're randomly dispersed because the way you generate this chip with DNA is actually randomly attach DNA molecules to the surface. And so what happens is every cycle you scan the surface, you collect this image, you process it, convert it, digitize it to sequence data, and then at the next cycle you come back and you recognize each spot, and the color may stay the same if it's another one of the same letter, or it may change. And so you build up a sequence at each of these spots. And you're doing the whole surface in parallel. Now, we don't know what the fragments are. They're randomly generated and they are randomly dispersed on the surface. So how are we going to build up a genome from that? Well, the Human Genome Project was going well, and we were sure that this reference would be generated. Now, early on, we did some calculations that convinced us, although the world wasn't convinced at that time, that if you could read at least 25 continuous bases of a randomly generated fragment from the human genome, even if there were one or two what we call SNPs, single base polymorphisms or mutations, you could still use a computational aligner to find out where it came from by comparative analysis with the reference genome. And so how this process works is you sequence many, many fragments of DNA, and then you use a computational aligner that starts scanning the genome to find out the nearest match of where it came from. And then the fragment sits there. And you oversample, so you sequence each part of the genome many, many times, and you stack these reads. And you can go down and you can identify the base at that position. And you can tell whether it's the same as a reference or whether there's a variation. Now in fact, today this process works well enough to get several hundred bases of reads. And so this process of aligning is much more accurate. And so by doing this, you build up a picture of the whole genome. Okay, so we formed a company, Solexa, to hammer this out into an integrated working technology. And actually, the reason we formed a company was twofold. In the early days, it was just David, myself, Colin and Mark tinkering in the lab, exploring each of the areas that we felt needed to be solved to bring this to life. Then we realized to really generate a system, we needed chemistry, we needed molecular biology, we needed software, we needed hardware engineers. We really needed to build quite a big interdisciplinary team. And at that point in time, there was no way of doing this in a university. So we actually raised money from investors to build a company to assemble a team to reduce this to practice. The other thing was we were both physical scientists, not geneticists. And the only way this could really be useful and have a chance of generating impact was if we could build a system that worked well enough to be able to put it into the hands of people who knew what to do with it. So in 2006, so this was really 8 years after we started the project, I thought it was a long time at that time. I was a lot younger then. I now look back, and 8 years is a blink of an eye in science. Actually, it was pretty rapid. We actually produced this commercial system, and it was called the 1G genome analyzer because it could sequence a gigabase, 1 billion bases of DNA in one experiment, which coincidentally matched that calculation we made in 1997. Some of the dimensionality of that calculation turned out not to be well matched, but the order of magnitude was matched. So this was actually quite a breakthrough at that time, and to put it into context, one of these machines had a capacity roughly equivalent to the global capacity as it was when we started the project. All these genome centers in one box. Now, in January 2007, an American company called Illumina acquired the technology and the company, and I stayed involved with them for another 9 years. And they optimized, engineered, improved on the technology, and they're still doing that today. And they produced this instrument in 2021 called a NovaSeq. Some of you may have seen one of these. Now, one of these instruments could sequence several trillion bases of DNA in one experiment, push of a button. And this sort of represents the global capacity as it was when these things were around. So it's another 3 orders of magnitude improvement. Now, there are production genome sequencing versions of this, and effectively they sequence the equivalent of about 1 genome an hour at very high depth, 50-fold depth. 1 genome an hour, 1 instrument run by a fraction of a technician. I haven't mentioned cost. Now, cost, there's cost and there's price. Price is more of a commercial thing, and cost is actually what it really costs. But even the commercial cost today is quoted at around $200, and I think it's pushing towards $100. So cost, capacity have changed by, it's actually over 1 million-fold in a period of about 20 years. Now, our goal, you'll remember, was to enable population scale sequencing, because we felt this had to be enabled to unlock what the human genome has to tell us. All right, so this is the giant panda. I show this because we get obsessed with human beings, and of course human beings are sometimes part of the problem with the world. But genome sequencing, of course, applies to plants, other mammals, fish, bacteria, viruses, and so forth. So there has been a sort of an explosion in understanding the genetic characteristics of other organisms. And the panda genome was sequenced using this approach. Now, in 2001, saw the release of the first draft human genome from the International Human Genome Project, genome number 1. 2008, there was one edition of this journal with 3 human genomes. At this point, there were 3 human genomes in the database, doubled to 6 with just one edition. One was the first Asian genome that was sequenced at BGI. One I was involved with was the first Yoruba African genome, and the other was actually the first genome of a cancer. So one edition tripled the number of genomes sequenced. Now, some of you may have heard this term personalized medicine or personalized genomic medicine. What does this mean? Well, I'm going to give you some examples to finish off with of where we are currently in this sort of promise or vision of unlocking useful insights about human health and what we can do about it from genome sequencing. Now, sequencing on a population scale is here and now. A lot of this is embodied in large-scale projects, such as the International Cancer Genome effort, and also state-funded projects, including in Singapore, there's a project to sequence 30,000 genomes. In the UK, the state-funded healthcare system, the National Health Service, has a project embodied in an organisation called Genomics England. Initially, it was to sequence 100,000 patients as part of their care. It's been extended to 350,000. This was actually initiated by David Klenerman, the then Prime Minister at the time. There are other equivalents. FinGen is in Finland. In the US, this project is going to sequence 1 million people over a 10-year period. Now, part of what these population scale projects are doing is sequencing, and over 1 million people have been sequenced now. There's another project called Biobank in the UK, which is 500,000 people have been sequenced. Is to sequence genomes, monitor the health of these individuals, monitor their lifestyle throughout their entire lives, and to build up really quite complex aggregated data sets that can be interrogated. These are typically made publicly available so that data scientists, computational scientists can interrogate it and look for relationships. So this is an ongoing experiment. Now, what can you do with knowledge about the genome? So I'm going to start with cancer, which of course is a multitude of diseases. Cancer is caused by changes to your DNA at the molecular level. Therefore, it follows naturally that understanding the genome and understanding the genome of cancers is going to tell you something important about what type of cancer is it. Now, I'll illustrate with one example. This is one that was found quite early on. BRAF is the name of a gene. It's a receptor signaling protein encoding gene. And this is a particular, all you need to know is that this is a mutation in this gene that occurs commonly in cancers. It occurs very commonly actually in skin cancers, melanomas. And in cancer patients that have this particular mutation, there is a drug that works against the protein encoded by this gene that is really, really effective only in those patients. Much less effective in patients that do not have that mutation. So this is an early example of genetic information telling the physician something important about the tumor, which allows a very precise prescribing decision to treat the cancer that's much more effective. Now, projects such as this, ongoing projects, are looking at patterns of genetic changes and taking those patterns to subset cancers and cancer patients. And in many clinics now around the world, it's become a standard of care to have your cancer genome sequenced and look for differences. And in some cases, there's enough information to help the physician treat the tumor optimally. But we're in the early phase of this. I think over the next 10, 20 years, as the sort of experiential information accumulates, we'll find out the full potential of genomic medicine in cancer. Now, diagnosis is also undergoing a potential transformation at the moment. I'm going to highlight one aspect of early cancer detection that's being explored and clinically trialed. Now, this is based on the fact that tumor cells tend to die, they necrose more rapidly than healthy normal cells. And when they do, they spill their contents into the blood. And it has been noted that you have fragments of DNA floating around in the blood with no cells around them. And it's been shown that some of this DNA is derived from the tumor. So if you take a blood draw, a few milliliters of blood, spin it down, isolate the plasma, isolate these fragments of DNA, sequence absolutely every fragment of DNA that's there. You can look for genetic patterns, and actually there's another chemical aspect of DNA that we call epigenetics. So you can also detect epigenetics by sequencing. And you can detect information that tells you there's a tumor. And even in some cases what type of tumor it is and where the tissue of origin is. So there is a clinical trial ongoing in the UK, in the NHS, by a company called Grail, with 140,000, not patients, people, scanning them for 27 different cancers using this approach. So the results are continuously coming out, but the vision here is to try and see through blood-based screening whether cancer can be detected earlier. And early detection for many cancers is absolutely night and day in terms of patient outcomes. Another area I'll briefly touch upon is, how am I doing for time, by the way? 5 or 6 minutes, all right, good. There's a sort of class of condition that generally afflicts young children called rare diseases. They're called rare diseases because most of them don't have names. And it's just really difficult to characterize them. Now, 1 in 17 people have a rare disease. So collectively, they're not rare. But there's a sort of uniqueness. Many of these cases, there's only 1 or 2 that's ever been found in the world. Now, rare diseases are often genetically caused, changes to the DNA. And they manifest as developmental disorders. Now, typically what can happen in a lot of pediatric clinics is mum and dad walk in with a child who has obvious problems, developmental problems. And there's test after test after test, often only informing the parents what the child does not have. So projects like Genomics England, and there are many others around the world, started sequencing the genomes, often what we call a trio. Sequence mum, sequence dad, and sequence a child. Because a child's genome is some combination of mum and dad. And then they can rather quickly identify those features that are unique to the child and absent in mum and dad. Because most of the genome will be the same. And in quite a high proportion of cases, I think the figures are between 25 and 30%, they are able to pin down the causation of the disease using this approach, where previously there was no diagnosis. And in some of those cases, there's an immediate treatment that's suggested. A lot of these turn out to be metabolic diseases, where nutritional changes can actually go a long way towards helping the child develop normally. In other cases, there's no treatment yet, but once causation and mechanism is understood, there's a basis for then developing therapies and treatments. Very first example of this that I was aware of was carried out by a pioneering chap called Stephen Kingsmore in collaboration with Illumina Cambridge. And the samples came over to Cambridge, mum, dad and child. They were sequenced, annotated, sent back to the physicians. They triaged the data, made a diagnosis, and it was treatable. And this was all done in 4 days. Now, Kingsmore is in the Guinness Book of Records now because he's done it in 24 hours from blood sample to diagnosis. The last area I'll briefly touch on is infectious diseases that are caused by pathogens. Pathogens have genomes. Sometimes it's an RNA genome. I'm going to take the case that's afflicted us all recently, and that's the pandemic that started in late 2019. So the SARS-CoV-2 virus was identified in Wuhan by next-generation sequencing, actually, using a technique called metagenomics. Now, metagenomics is a process of taking samples from the environment or clinical samples, not purified, so there can be all kinds of DNA in there, and you literally sequence it in great depth, and then computationally assemble genomes, plural. So this approach was what identified the SARS-CoV-2 virus as a new virus. Then, as the outbreak started to spread and variants started to emerge, patients were having not their genome, but the viral genome sequenced when they tested positive. And there was a database set up, a real-time database that had over 1 million cases being updated continuously. And so there was a global geographical map of the spread of the virus, the emergence of new variants in real time. Now, this was very helpful and a sort of first case example of doing epidemiology in this way. It helped public health controls. It also actually informed about the sequence of the target sites for vaccines. And as you know, mRNA vaccines were transformative during the pandemic, and they're still being administered. But knowing the sequence of the virus and variants of the virus allows a very logical and direct optimization of the mRNA sequence to provide the very best vaccines. So the future may well be blood-based surveillance of pathogens and also internal diseases in people, right? That's one version of the future. So I'm going to end with this slide. So many people to thank from the university, Solexa and Illumina. I'm just going to leave you with these thoughts. Thank you very much.
Thank you, Sir Shankar, for sharing with us your insights. May I request you to take a seat for the Q&A session. I would now like to invite Professor Tan Meng Hao from the School of Chemistry Chemical Engineering and Biotechnology, to be the moderator for the Q&A session.
All right, so thanks, Shankar, a lot for the fascinating talk. So any questions from the audience? Anything you want to ask about DNA? Yes, there's a question there.
Hello, Professor. As you said, the DNA, the work on DNA, like sequencing it, is a large work and it needs a lot of time. Although you have some new machines to help you work with it, but I want to know, have you thought about, or do you work with, like, add AI into your work to help you like work faster, or they can maybe do something else to help your research?
So I don't use AI directly, but I think certainly machine learning, so Bayesian machine learning, has been used to essentially do pattern recognition in complex sequence-based cohorts for the purpose of developing and optimizing diagnostic signatures. And so I think that has been going on for some time, I think with some success. I think where AI has huge potential is in those large-scale programs that I described, where there's sort of long-term follow-up on population-scale cohorts of people, looking at their lifestyles, their healthcare records, their exercise regime, and so forth, having measured not just their genome, there are other biological measurements that are made as well. So that's a very complex, multidimensional data set that's probably quite difficult to model using some rational mechanistic hypothesis. So I think the scale and complexity of these data sets is very likely to benefit from unbiased AI-type approaches to pick out patterns that are important.
Hi, I'm Joanne. I'm Associate Professor of Genomic Medicine here. So I use genomics on a daily basis. So firstly, to thank you for the fact that now we have easy access to genomics for our patients. But two questions, if I may. The first one is, when I first came back from the US from training and I was starting to use genomics in the clinic, it struck me within 2 days that I was actually making the problem worse because of the social cost of genomic testing in the region, especially in Singapore and in Asia. It's an out-of-pocket expense. And on a daily basis, I was practicing very inequitable medicine, given that people who could afford it were able to pay for information, and people who could not afford it still remain. And so this work still continues, and I just wanted to hear your thoughts of how you actually make this really affordable for the region. Because when I travel to Indonesia to train the folks in Indonesia about genetics, I'm almost worried about doing it, because it really does then lead to the people who could afford the testing getting the pre-information, and the people who don't not having it. So that's the first question, sort of a bigger picture kind of viewpoint. The second one is just more for the students. Have you had your genome sequenced? And if not, why not?
Both really good questions. First question first. You're absolutely right that genomic medicine is a first world thing at the moment. Frankly, as is a lot of medicine. The very best cancer treatments are, some of them are only for the elite first world citizens, not even everyone. So this is a problem that needs solving. So how are we going to solve this for genetically based diagnosis? So I have two thoughts on this. One is, this approach is industrialized now, and I think it's still responsible for about 80% of the sequence information. And the volume is increasing. So volume increases, help drive the viability of cost reduction. And we've seen more than 6 orders of magnitude of cost reduction in 20 years. And I think it's part technological, but partly a commercial question as to how low can that go. And I believe the genome will drop below $100 per genome. Now, maybe that's not low enough for some populations. We'll see how low it goes and whether it can be truly democratized. But I think the other part of this is many of the population scale studies are a combination of real-time treatment and learning. And so most diagnostic tests that are genetic actually just use a rather cheap PCR-based test, because once you know where to look and test, the cost goes down another 2 orders of magnitude and becomes affordable anywhere. So I think in this sense, the countries and organizations that can afford to fund the experiment do. And everything is, the nice thing is, everything's made very public. The data can be mined by anyone. So I do think cost-effective testing will ultimately be democratized, because you don't necessarily have to sequence a genome to do genetic testing. Now, second question, have I had my genome sequenced? Short answer is yes. Actually, I had my genome sequenced about 12, 13 years ago. And partly because I wanted to understand what it felt like to go through the process. And there are many size sequences, and even the small ones are good enough to sequence a genome now, but actually you're not allowed to. You need to go through an ethical consenting process. And certainly in the UK, if someone wants their genome sequenced today, there has to be a clinical rationale for it, and it has to be done through a clinician who orders it like a test. So I went through that with a clinician, and I had to go through all the implications of what do you want to know, what do you definitely not want to know. And I was made to think about that through having my genome sequenced, I was inadvertently having half of each of my children's genomes sequenced. And so I had to go through the thought process of what if I'm okay, but one of my children isn't? And so that was my rationale. So what did we find out? So when the results came, the first thing my geneticist clinician said is, gosh, you have a lot more variation than normal. And I said, well, that's because the reference genome is of a white Caucasian genome, and I'm not that. So most of these variations are to do with ethnicity. It actually highlighted a problem in that there is no Platonic genome. We need reference genomes that reflect the population in different parts of the world, and that's starting to be addressed. Otherwise, there were only sort of 3 literature-based things that popped up. One of them, he said, if you had any problem, you would know by now, so you're probably fine. The other one, he said, I don't believe the literature, so ignore that. And the third one, he said, is you're predisposed to hearing loss. And I said, well, my wife could have told you that before I had my genome sequenced. So, but once you're sequenced, you get updates. It's rather like a software update as new information is found. So it was an interesting experience.
Good afternoon, Prof. Thank you for the great presentation that you bring. So I'm actually kind of like, I don't know anything about this field, but I am kind of interested in what are the social implications that will happen if you try to sequence a lot of people's genome in the world. So I heard that, let's say you want to test a group of population in a city, then you will get the data and so forth and so forth. So first question is, what are the barriers that currently has emerged when you try to sequence the genome of these people? And the second one, since you said that the data is public, is there like any kind of bad phenomena that's happened because of some people using the data wrongly? Maybe that's it for now.
Thank you. It's an excellent question, an important one. So, I mean, firstly, you can't just have your genome sequenced. You have to go through a consenting process. And so, typically, there has to be a clinical reason today for you to have your genome sequenced. Now, these projects, like I described a project called Biobank in the UK, which is 500,000 volunteers who've given biological specimens that have been sequenced, and other things have also been measured. Now, this is really a discovery endeavor that each of these individuals is consenting to enabling. And the first thing that happens to samples is they become anonymized. So you can't look at the data and recognize that you're looking at someone's data. Okay, and this is, of course, it's done with clinical data anyway. So I think the individuals are as protected as computerized systems can be protected and anonymized today. And as I said, people have to consent to these processes. So I think the potentially more controversial thing is the use in forensics. So genome sequencing is now starting to be adopted in forensics. And previously, DNA fingerprinting was used to take a sample and see if it was a match. I think the difference with forensic use of whole genome sequencing is if there's DNA left at the scene of the crime, you can actually pull out a lot of characteristics of the individual. Ethnicity, eye color, hair color, probable height, etc. In fact, there are even some attempts to paint an image of what the facial features of the individual might be on the basis of their genome sequence. So some of this may pop into forensic use. And the idea of the DNA database for criminals is it's also a controversial topic. So I think we haven't yet fully ironed all these things out, but there are social implications for those. But I think data and privacy is rather like other aspects of data and privacy that we encounter. So the data is anonymized.
Just curious to follow up on two of those ethical aspects. You said there has to be a clinical reason, but there are lots of companies now, ancestry companies and things offering sequencing. Even reputable companies like National Geographic will, you spit on a card and send it off and they tell you you're 80% Norwegian or something. I don't see how that correlates with what you said about there must be a clinical need. And then the second point, I can understand what you're saying about anonymizing the data, and that's true of finding an individual. But supposing the sequencing showed that a particular ethnic grouping had a predisposition to certain diseases, and therefore insurance companies said, oh, he's from this ethnic grouping, they pay 3 times as much for their health insurance as somebody from this ethnic grouping. That starts to get a bit delicate in terms of, the morals of doing sequencing and what happens to the data. So I just wanted your thoughts on those two.
Yeah, no, two very good examples, actually. So the Ancestry.com, 23andMe, these consumer type companies, I mean, firstly, they're not sequencing. They're doing something called genotyping, which means they're sampling certain spots in the genome using an array-based method. So it's not whole genome analysis, but it still does give genetic information about the individual. And these companies formed out of the blue, and the system wasn't really ready to deal with this. And particularly one of them is also a sort of social networking company as well that allows you to find your genetic look-alike friends and people who may have a genetic predisposition to the same likings as you. Now, this was all done as fun, and I think the individuals have a say as to what happens to their data. I believe it is possible to simply say, give me back information about what flavor ice cream I should like, and etc. All these fun things without sharing my data. I think you can stop it from being pooled. But I think most people want it to be pooled because then they have this network effect. So people are making a choice there.
And not a choice for clinical reasons.
No, that's not a choice for clinical reasons, and they are actually not allowed formally to give back any information that's clinical. So it has to be these other lighter things. But I think inevitably, and this has already happened with one of these companies, where the FDA has written to them and said, actually, you've crossed the line by giving information about this or that. So that's still a grey area that you've quite rightly spotted. Now, your second question, sorry, remind me again.
You spot within the database, the public database, you can't identify individuals, but you identify an ethnic trend, a predisposition towards certain types of cancer,
yeah.
Then you can imagine insurance companies saying, we're not insuring this group of people.
So, I'm ethnically South Indian, which means I've got a predisposition to coronary heart disease and type 2 diabetes. So that's already factored in. So if I take out medical insurance, I'll declare that I have a family history, but Actually, I don't need to. I'm not going to defend insurance companies, but what they do is risk management. And so they already ask us questions that can lead to conscious biases, some coincidentally linked to ethnicity or otherwise. So I think that genie's already out of the bottle, actually. But in the UK, certainly, I think, so I've had my genome sequenced. I'm under no obligation to provide any aspect of that information to any insurance company. So there is legislative protection at the moment, specifically on genome sequences.
Very good, thank you.
All right.
All right, very interesting questions on ethics and privacy and so on. But this debate will go on for a very long time. So for the sake of time, we have to stop here. So let's thank Shankar for the talk again.
Let us put our hands together to thank Sir Shankar and Professor Tan.